I think we'll go ahead and get started, since we're starting a little bit late. I'm Emery Brown. I'm from BCS, Brain and Cognitive Sciences, Picower Institute. I'm going to be the moderator for this session on brains. So why think about the brain? It's this fantastic organ. It only weighs 3 pounds, but it consumes almost 20% of the oxygen in the cardiac output. And studying the brain is a challenge but what's cool about it is that everything we learn can help us with new diseases relevant to the brain. And the critical factors, as was mentioned earlier in today's first session, is that just like the other areas of investigation that were discussed, brain research and brain-related improvements in healthcare, there are 3 things that converge here at MIT: advances in science, engineering, innovations, translational applications. And you'll hear that today from our speakers. So these are our speakers. We're very happy to have Ed Boyden from the Picower Institute, Mark Baer, also from the Picower Institute, Josh McDermott from the McGovern Institute, and also Evelina Fedorenko from the McGovern Institute. And we just made a little chart to show you that indeed these 3 areas will be touched throughout with the talks that you're going to hear. So with that, I'll turn it over to my colleagues. Ed.
All right. Yeah, thank you so much. This is really a fun day, and it's great to see so many convergent fields in this amazing and important area. I direct a group that tries to confront, through technology, the spatial and temporal complexity of the brain. And this is a great arena for innovation. I mean, the brain is a gigantic object, right? And brain cells are gigantic objects. But the wiring of the brain is nanoscale, right? And the wiring is extremely tiny. And the connections between brain cells are also very, very small. Also, we have molecules, right? The human genome has what, 30,000 or so genes encoding for countless biomolecules. And we need to know what they do and how they work together and how they go wrong in disease states. But it's even more complicated because there's the time dimension too. You know, if you think about Alzheimer's disease or learning or aging, almost any process that's important to us as people, there's an extended component of time related to these functions. Yet the elementary neural codes of the brain are very, very brief, millisecond time scale electrical pulses. So we build tools to try to help neuroscientists conquer these differences in space and time. Now, why is it important? Well, lots of technologies exist. On the left, MRI scans. On the right, microscopes. But it's very hard to image a large object like a brain down to those nanoscale dimensions. So in our group, we practice a lot of creativity skills, which we think can be learned and practiced. And one idea is to do the opposite of what people are doing. And for literally 300 years, the way that biologists magnify images of cells is with some kind of lensing effect, right? We started wondering, what if you could magnify cells directly? And so the basic idea we call expansion microscopy. We chemically install a dense spider web-like mesh of basically baby diaper polymer inside the brain. Not a living brain, but a preserved one. And if you do it just right, you can weave that chemical mesh inside cells and outside cells, around biomolecules, in between biomolecules. And you can magnify the brain physically. So in panel B here is a piece of the mouse brain, several millimeters on the side. Panel C, the same piece of brain tissue, about a day and a half later, but we've blown it up by 100 times in volume. And the polymer starts out super dense, like in the upper left in that cartoon, and ends up like in the lower left. Here's a little movie of a piece of brain being expanded. So we formed the baby diaper polymer earlier, and it's half an hour sped up to about half a minute. But we add the water right there. And I hope you can see that this piece of brain tissue, which is permeated by the baby diaper polymer, is physically growing, right, before your very eyes. That's great. So now we can use imaging tools and see tiny, tiny things that are too small to be seen by regular technology. And that's important because, again, molecules are tiny, and the wiring of the brain is also really tiny. And yet you want to understand it across an extended 3D spatial extent. So here's an actual piece of brain tissue. This is from the mouse hippocampus, which is involved with many functions, including related to memory, but lots of other things as well. And the brain cells are expressing fluorescent proteins. So these are proteins borrowed from jellyfish, coral, and other creatures. And it gives you a color code or barcode, if you will, so you can tell the cells apart. We deliver the genes for these proteins using viruses. We then preserve the brain and expand it using the method that I told you about. And now we can see the fine wiring much better than before. So we're going to fly through this piece of brain right now. And I hope you can see that these individual processes, which have distinct colors, are much, much easier to see than one might guess. And then, of course, we can zoom in and out, right? It's intrinsically a multi-scale way of visualizing the brain circuitry. So we're very excited about this idea and how we can use it to map entire brains. But of course, this doesn't work on a living thing. And so we have to really switch gears here to tackle the second question, which is time. How do we control these fast electrical pulses of the brain? If we could control them, then you could drive certain brain cells and figure out what they do, right? Do they trigger a behavior or a pathology or perhaps the remedy of a pathology? And so the idea that we brought forth is this concept that we now call optogenetics, because opto is about light, and genetics is because we use genes to mediate light sensitivity. The basic concept is basically like this, right? Suppose you had solar panels that convert light to electricity. If you could somehow install them on brain cells, shine light on the solar panels, convert to electricity, you could control the brain. And then you could bring light into the brain. The electrodes are brought into the brain all the time, because it doesn't feel pain. We can bring optical fibers in as well. So how do we do it? Well, we have to make the neurons sense light, which means we have to find those solar panels. And it turns out all over the tree of life, you can find molecules that do that, right? So here we are looking at a single-celled green algae, and it has an eye spot in the back there, which helps it sense light to control those flagellae, those tails that help it swim. We just zoomed into the eye spot. And it's chock full of these proteins that convert light into electrical signals, ion fluxes, right? Kind of like those solar panels that we were just talking about. So what we can do is take advantage of the fact that this is a protein. It's genetically encoded. It has the right temporal and other properties, right? It's a fast response to light because you just open up a pore and instantly get ion flux across. And so the idea they brought forth was, let's borrow this gene and see if we could put it into the brain. And so this is where we had to just sort of count on serendipity. We're very excited now, by the way, to try to use AI and other methods to do on purpose what we did accidentally here. Can we go out and look for treasure in the natural world and not just, wait around for it to show up? Anyway, we can take the gene that encodes for this light-activated protein. We can install it using a gene delivery method, like a gene therapy vector, into the brain cell. The brain cell will manufacture the light-activated protein, and then again, serendipity kicked in again. It was safe. It worked. It didn't have to work, right? The algae could have been a happy home for the protein, and the neuron could have died or malfunctioned. Anyway, it was really surprising in a pleasant way that when we shine light from a laser or an LED on these brain cells, they fired electrical pulses, not unlike the ones happening in your own heads right now as I say these words. So there's lots of things you can do. You can take different parts of the brain, put these light-activated molecules in them, try to see what happens, right? So I'll just pick one example because it has a nice movie, but dopamine is a very important neuromodulator in the brain. In the popular press, it's often called like the pleasure center of the brain. Could you find out whether reinforcing the activity of these neurons also reinforces behavior? And so it's a very simple experiment here. Mice go and poke their nose in a little portal, and if they go to the right one, they get a pulse of light that aims at these dopamine neurons, which are expressing the light-activated protein. If they go to the left portal, nothing happens. And so you can see the result. Pokes his nose, gets a pulse of light, does it again, does it again. And so this mouse is basically working for light. So this is very widespread. We share all of our tools freely, by the way. So expansion microscopy that I talked about before is in use by thousands of research groups, almost 900 experimental results coming out already. Optogenetics is in use by thousands of research groups as well. And interestingly, these molecules are starting to be used in people as well. So many people are blind. They've lost their light sensors in their eyes. A team in Europe got one of our molecules, discovered here at MIT, and was able to deliver the gene for it into the eye of a person who lost his normal light sensors. And they converted the spared cells of the eye into, well, kind of a camera. And so he was able to recognize household objects. He could see doors on a hallway. Not perfect vision, couldn't recognize faces very well, struggled to read text, so more work to be done, but still, a very interesting advance in an otherwise extremely difficult to confront condition. Going forward, we want to integrate these tools. We've always shared the tools freely, but we want to start combining them towards this goal of making computer simulations of the brain. If we could simulate the brain, of course, that'd be very interesting from an AI perspective potentially, but also we could maybe look under the hood and poke around in silico, in software, and try to figure out where in a network to intervene to help with the disease state. And of course, if we could simulate brain computations of different kinds, it could become a tool that we could use to probe different scientific questions. So in the coming years, our hope is to start with small brains like fish and worms and start going to larger and larger brains. And this is sort of a complicated slide, but it sort of outlines one of our ways of trying to put together all the different approaches that we take. So the upper left, we want to look at the brain while it's in action, the blue part. In the lower left with yellow, we're going to use the expansion method to make maps of the brain. And then the three blocks on the right are ways of making different models, machine learning models, biophysical models, and so forth. So to conclude, we're very excited about these tools we're building to see, map, control, and understand the brain. We share them all very freely, but in the time to come, we're very excited to try to integrate them into a workflow that hopefully can help us build computational models as well that can be of great use for others. So thank you.
So I'm going to grab my jacket here.
All right. Sounded like an unruly classroom earlier, but I'm glad everybody's calmed down. A sad fact of life is that diseases, deprivation, and injuries can impair brain function, and it's extremely difficult to restore brain function, particularly in adulthood. And a very classic example of this is a condition called amblyopia, which is a condition that it's a visual impairment that develops in children that don't have high quality vision during infancy. And that's what's shown here on the left side. This is a child that has a cataract in one eye. The cataract's easily detected and can be surgically removed and replaced with an artificial lens. But as a consequence of that altered visual experience during infancy, there's a permanent visual disability. So there's a very severe visual disability in the amblyopic eye, the eye that had been deprived, and a loss of stereoscopic depth perception that has a major impact on the child's life. Now, there is a treatment for this type of disease. It's called patch therapy. It was introduced well over 100 years ago and is still the medical standard of care, which is something of an embarrassment, I would say. But patch therapy can work, but so the patching occurs in the other eye, which we call the fellow eye, and it forces vision through the eye that had been deprived. The limitation of patch therapy is that it only works very early in life. In this case on the left, in humans, if the patching is not done before the age of 4 months of age, then there is no recovery of vision. So if the cataract was not detected early enough in life and corrected, there's really a very poor prognosis for that child. We've been modeling this for over 50 years in animal studies, and that's what's shown on the right. It's just a similar story. This is a cat model of deprivation amblyopia as well. So I didn't mention, but the cause of amblyopia is an impaired synaptic development in a part of the brain called visual cortex. So what we'd like to do is to harness the principles of experience-dependent synaptic plasticity to promote recovery of brain function, particularly in adults. So over, again, the last half century or so, we've learned a tremendous amount about the mechanisms of synaptic plasticity at excitatory synapses in the brain. We know that synapses are bidirectionally modifiable. And one of the critical triggers for this plasticity is a neurotransmitter, whoops, can I go back one? A neurotransmitter receptor called an NMDA receptor. And what we've learned is that activation of NMDA receptors can trigger both synaptic depression and synaptic potentiation. And the critical variable is the level of postsynaptic response at the same time those receptors are activated. And so what you see on the right is a function where we can map the consequences of NMDA receptor activation depending on the level of postsynaptic response. So there's a critical site here. Again, go back, please. My laser pointer doesn't work. One forward. Thank you. So you can see strong NMDA receptor activation can yield a synaptic potentiation, and weak NMDA receptor activation yields synaptic depression. So what is controlling this postsynaptic response? Well, obviously, it's how many synapses are active at the same time and what the strength of those synapses is, and also the concurrent level of inhibition in the brain. So we'd like to understand how we can promote the conditions that will lead to synaptic strengthening. So a lot of our work was influenced by a theory of synaptic plasticity that was introduced by Leon Cooper. Leon was a mentor, friend, and colleague of mine. Sadly, he died just a few weeks ago. He's a very famous physicist. He won the Nobel Prize for a theory of superconductivity. But after that, he went on to become interested in synaptic plasticity in the brain. And he developed a theory that is now called the BCM theory, or Bienenstock-Cooper-Monroe theory. It's a very simple idea. And what he proposes is that the synaptic strength varies as a function of the product of the input activity and a concurrent level of postsynaptic response, some function of the concurrent level of postsynaptic response. And that function is shown here in the middle, and it should bear a close resemblance to what I just showed you. In fact, Leon's theory inspired the experiments that revealed the properties of bidirectional synaptic plasticity in the brain. A critical variable in Leon's theory was that crossover point between synaptic depression and potentiation doesn't have a fixed value, but rather it's free to slide back and forth depending on the average activity of the cell. So in other words, the rules of synaptic plasticity are themselves plastic, and that's a property we now call metaplasticity. So the question is, can we slide that threshold to the left to promote synaptic potentiation and allow recovery of function? Next slide. I guess I can do that. OK. So the way we addressed this was to take advantage of a neurotoxin that is derived from pufferfish. If you've ever had fugu, it's a delicacy in Japan. It's a type of sushi that is prepared from pufferfish, but it can be quite dangerous because in the liver and ovaries of pufferfish is a lethal toxin called tetrodotoxin. The reason it's lethal is because it blocks all action potentials, it blocks all nerve impulses. It can be used in experiments to block electrical activity in the brain. So the experiment that we conceived was to inject tetrodotoxin into the eyes of an animal that had been rendered amblyopic, shut off the activity of the visual system, allow that threshold to slide to the left, and then restore vision and ask, do those synapses regain strength? And I wouldn't be telling you this if it didn't work, and the results were really quite dramatic. So this is experiments that were done in a cat model of amblyopia. We've done the same thing in mice as well. And the graph on the left here shows the acuity of either the amblyopic eye in blue or the fellow eye, the good eye, in green. And so this animal had been deprived of vision in one eye, in the amblyopic eye, for a period of time. That eye was opened. There's no spontaneous recovery of vision, just as the case in the humans. But at the time indicated with the TTX and the downward going arrows, this drug was injected into the eyes. And then, of course, that shut off all the activity in the fellow eye. But now we wait and watch what happens. Okay, so when the TTX wears off, the fellow eye response returns to normal. But amazingly, the amblyopic eye response comes bounding back and goes right up to a value that's comparable to the fellow eye. So a complete reversal of amblyopia. And this experiment was done at an age when traditional therapy is no longer effective. So that's what's shown on the right side here, that downward going arrow, that this is occurring out at about 14 weeks of age, which is well beyond that sensitive period when patch therapy works. So we were very excited by these results. It seemed it was a theoretically inspired study, but it really was based on an assumption, and the assumption was that TTX actually shuts off activity in the visual system. However, more recently, we discovered that if we can shut off activity in the retinas, but the relay nucleus that brings information from retinas to cortex is not quieted at all. In fact, the activity in that relay nucleus, which is called the lateral geniculate nucleus, that activity starts to fire off in bursts, spontaneous bursts, high-frequency bursting activity. And that bursting activity occurs both in the neurons that are postsynaptic to the inactivated eye and to the other eye as well. So it's a qualitative change in the type of activity that's going to the cortex. So the question then became, geez, are these bursts really required for this recovery? So I have a wonderful graduate student, Maddie Ekavari-Leet, who tested this by knocking out the gene that was responsible for this bursting activity in the lateral geniculate nucleus. She can eliminate bursting, and when she did so, she eliminated the recovery that was caused by the TTX injection. So we have to really rethink what the mechanism for this recovery is. And I don't have a good answer for you, but what we do know is that the bursting activity sets up a pattern of very highly synchronous activity in the brain, and also impairs inhibition in the cortex. So we think we've achieved what we hoped to achieve, not by sliding the threshold necessarily, but by evoking a stronger postsynaptic response. So the mechanism is still obviously under intensive investigation in the lab, but we can already start to think about how we might apply this in a therapeutic setting. So the next step for us was to go to a primate model of deprivation amblyopia. So we've repeated these experiments now in monkeys that were rendered amblyopia by early life monocular deprivation. And the results are, again, very dramatic. So we can get a very substantial improvement in an electrophysiological measure of visual function in monkeys. But this also, there's been, nature has provided this experiment in humans as well. There are amblyopic humans that, unfortunately, have suffered damage to their fellow eye in adulthood. So they have relied on their fellow eye for vision for their whole life, but then because of injury or disease, the fellow eye has to be removed or is otherwise incapable of supporting vision. And what's observed is that in many cases, there is an unexpected recovery of vision in the amblyopic eye. So this means that that plasticity is there in our brains. We just need to find a way to tap into that. Now, losing the fellow eye is not a therapeutic strategy to promote recovery from amblyopia. But what is temporarily inactivating that fellow eye and essentially rebooting the visual system. So we think there's an exciting future ahead. And with that, thank you very much. And then our next speaker is Josh McDermott.
Greetings, everyone. All right, so we rely on our senses to perceive the world around us, but as Mark just told you, our senses are fragile. So in fact, over 39 million people worldwide are blind, and over 70 million people worldwide are deaf. And we would love to be able to restore the senses in these individuals. Now, certainly the treatments for these conditions have improved over time, but we're far from being able to fully restore sight and hearing. All right, so the first thing you all need to know, you all have receptors in your ears called hair cells that are responsible for turning sound into electrical signals, and they're shown here. So they sit on top of a membrane in your ear that vibrates in response to sound, and the hairs on the cells get bent when they move around, and that causes changes in their membrane potential. And those signals are sent to your brain via the auditory nerve that's shown there. Now, deafness is typically caused by hair cell dysfunction. All right, the second thing you need to know is that in the last 5 years, there have been huge advances in our ability to build computer models of sensory systems. And this has been really largely driven by progress in artificial neural networks. So when we do this in our lab, we typically start with a model of the ear that's rooted in everything that we know from biology. And then we put on top of that an artificial neural network that gets optimized to do different kinds of auditory tasks, like recognizing words or localizing sounds. And what this long stream of fantastic students has shown over and over again is that when you build models in this way, they reproduce human behavior to a pretty remarkable extent across many different domains. Okay? And this has really transformed our ability to understand and study perception. So our goal now is to use these models to understand and improve bionic ears and eyes. So our lab studies the sense of hearing, so we're focused on bionic ears, but we think many of the insights will be equally relevant to vision. So the cochlear implant is a bionic ear. It aims to cause hearing via electrical stimulation, and it is the primary treatment for deafness at present. And there are over 1 million of these devices in use worldwide. Okay, so the way this works is there's a microphone outside the head, and the sound signal that gets recorded gets turned into electrical signals, and those are sent down that wire that goes into the cochlea and wraps around in that spiral. And the wire's got a bunch of electrodes on it. Okay? And so the key concept is that if you have hair cell dysfunction, that causes deafness. But if the auditory nerve that would normally convey signals from the hair cells to your brain, if that's intact, then that can be electrically stimulated, and that should cause you to hear something. So you bypass the hair cells to create the sensation of sound. Okay? So ideally, the electrical stimulation would replicate the nerve patterns of activity that would normally happen from hearing electrically. But that's really hard to do. And so although these devices are in some sense miraculous, they're still not close to restoring normal hearing. So one example of that is shown here. So I'll show you a number of graphs that will kind of look like this. So this graph plots how well people can recognize speech in noise. So the y-axis is word recognition accuracy, and the x-axis is the signal-to-noise ratio. So as you move to the right, the speech gets louder relative to the noise, and it gets easier to recognize. So the black curve shows how well people with normal hearing can recognize words, and when the speech is loud enough, they're at ceiling. The red curve shows what happens in humans that have cochlear implants. Okay? So on the one hand, the fact that they can score about 60% correct is amazing, because these are individuals who would otherwise be completely deaf. But you can also see that there's a large gap between the red curve and the black curve, and that's what we'd really like to close. Okay? All right, so what limits outcomes with these devices? Well, there's at least 3 potential factors that we know of. One is that the device strategies are almost surely suboptimal. Another is that if you undergo a period of deafness, the neurons in your auditory system die. Right? So there's neural degeneration to varying extents in the auditory nerve or beyond in what we call the central auditory system. Okay? And there's also the potential that your brain is suboptimally decoding the signals that it gets from these devices. Okay? All right, so what limits our research into understanding these factors? Well, one is that we have very limited access to what's actually happening in the brain of a human implant listener. So it's not like you can just look at a person and tell whether they have neural degeneration or tell whether their brain is suboptimally decoding those signals. Okay? But there are 2 other major obstacles. One is that the best outcomes typically occur after somebody has lived with an implant for a very extended period of time, months to years. Okay? And the other issue is that the outcomes are highly variable across individuals, as I'll show you in a moment. So it's not like you can get a new idea for a device, give it to one person, and then really extrapolate much about how it will work in general. All right, so this makes it very challenging to experiment with alternative device strategies. And so most of the major implant manufacturers have all pretty much been doing the same thing for the last 10, 20 years. All right, now what we can do nowadays is build very good simulations of the nerve responses that would result from a cochlear implant. And Anisha Banerjee is a fantastic grad student in our lab who has built one of these simulations. The details here don't really matter. It's a computer program that predicts the nerve responses from electrical stimulation. And so one example is shown here. So the top picture shows an example nerve response from a cochlear implant. The different rows represent different nerve fibers, and the x-axis is time. Okay? What's shown on the bottom is an analogous simulation from a normal ear to the same speech utterance. Okay? And so the point is that the cochlear implant is intended to produce nerve responses that look like what you would get from the normal ear, but you can see that they look really different. Right? And so that's probably part of the issue. All right, so how can we understand these outcomes with the models that we have of the brain? So one other important fact to note is that human implant users improve pretty substantially over time. So this is a really cool graph that shows word recognition scores as a function of the number of months post-activation, so how long someone has lived with their device. And each line is a different individual. Okay? The different colors don't matter. So you can see that all the lines go up, all right, and they go up over a period of time. All right? So people are learning to do something with their brains that makes them better at decoding the information they get from the device. But the other thing to note here is that the asymptotic performance is really highly variable. So some people end up doing great and others much less so. And so what we don't know is whether that variation is due to differences in how well people can learn, or whether, for instance, it might be differences in neural degeneration or some other factor that we don't know about. Okay? So these models that we build using machine learning, we think can help reveal the potential of brain plasticity. So we can take these models of the auditory system and build what we call a static model of hearing through a cochlear implant, where you take a model of the normal auditory system and you just give it input from a cochlear implant. But we can also build what we would call a plastic model, where you completely re-optimize the neural network to deal with input from a cochlear implant, simulating what would happen if your brain could completely readapt itself to this altered kind of input. Okay? We can also potentially look at the impact of degeneration by looking at what happens if you just delete a bunch of nerve fibers in the input to the model or delete a bunch of neurons. All right? Okay, so what happens? So here's one example result. Again, this is the same kind of graph that shows word recognition accuracy versus the signal-to-noise ratio. The black line shows a model of normal hearing that is very similar in its performance to humans with normal hearing. And the green curve shows a model that gets input from a cochlear implant, but that is fully plastic. So you can see that the green curve is actually pretty close to the black curve, right? It's a little bit worse, but really pretty close. Now the red curve shows the results of humans that have cochlear implants that we tested in our lab on the exact same experiment, right? You can see that they're not doing nearly as well as the model, the green curve that is, right? And so that suggests that in principle, you could do a lot better with the input from the implant if you could fully re-optimize your brain to process those signals. Okay? So can we simulate the possibility of incomplete plasticity? Well, one thing we can do is just re-optimize some of the parts of that neural network to deal with input from the cochlear implant. And when you do that, you get the blue curve. And that's now within striking distance of what you see from humans. Okay? Now, by contrast, I'm going to show you now what happens if you delete 50% of the neurons in these models. And those are the dashed lines. And the effect of that is actually remarkably modest. All right? So we see huge effects of the extent to which you can re-optimize your brain for this altered input, and really modest effects of neural degeneration. So it's consistent with the idea that plasticity is a dominant factor underlying outcomes, and it raises the possibility that increasing plasticity in the human auditory system, doing things like what Mark was just talking about, could potentially yield big benefits. All right, so the next steps that we're excited about are to use these models to derive new device strategies that could potentially further improve outcomes. And so we think the models can be very useful here in revealing the best case outcomes for candidate devices, potentially enabling large-scale screening. So you can try out lots of different ideas, and then the most promising options are things that you could potentially explore in humans. So I think this is a nice example of basic science with impact, where the models that we build of the brain and refining those models will lead to better therapies that we hope will help people. So thank you. And with that, it is a great pleasure to introduce the world's expert on the neural basis of language, my colleague, Professor Evelina Fedorenko.
All right, thank you. All right. So let's see if this works. So language is, I study language, and language is this incredible superpower we have. Using language, we can build deep relationships with each other and restore international political order. We can send submarines to the bottom of the ocean, rockets into space, and cure diseases. We can bring about political and social change and record the history of our planet and our species. So how do our brains achieve this remarkable feat? So using studies of patients with lesions, non-invasive imaging like fMRI, and intracranial recordings in patients undergoing surgery for brain tumors or epilepsy, we've learned that there's a whole bunch of areas in your brain that help you do this. And I assure you all of these areas are working in your brains as you're listening to me right now. These areas look something like this in any individual brain. And interestingly, they support both comprehension and production. This makes us think that these areas basically store our knowledge of language, so what words mean and how to put words together. And of course, we need this knowledge to both take a thought and encode it into a word sequence, or process somebody else's word sequence and infer the meaning that they intended. What I just said should make it clear to some of you who may have heard of things like Broca's area and Wernicke's area, that these language areas I mentioned are actually different from these areas classically discussed in the neurobiology of language. It turns out that these Broca's area and Wernicke's area are lower level areas. So Broca's area is an area that's important for planning speech motor movements, and Wernicke's area is important for perceiving speech sounds. So for example, if I now switch to talking in a language that's unfamiliar to you, your Wernicke's area will be working just as hard as it's working right now, because it's still getting its preferred input, which is speech sounds. But your purple areas, your language areas, the activity in them will plummet because you would no longer be able to extract any meaningful information from them. These areas, the language areas, are also distinct from areas that support thinking and reasoning, as my group and a few others have shown. Of course, our language system interacts closely with our reasoning abilities, but nevertheless, the language areas are not the areas that are doing thinking in your brain. And you can lose your language system, as in cases of severe aphasia, and still be able to think and reason in complex ways. A couple other things. The system seems quite robustly similar across diverse languages. There's over 7,000 languages spoken and signed across the world. We've tested this in about 50 languages, looks similar. For those of us who have multiple language systems living in our brains, like bilingual and multilingual speakers, they all live in that system. And developmentally, the system is there and shows kind of adult-like topography by about age 3. Now, our focus today is on health. So losing language is an incredibly devastating condition. Not having full access to language in development leads to all sorts of cognitive delays and difficulties in establishing relationships with your caretakers. And losing language in adulthood in cases of degeneration or stroke aphasia is life-changing in all sorts of horrible ways. So in trying to understand language development, we're trying to ask questions like, why is the language system sometimes doesn't develop properly, as for example in cases of autism? Can we predict language development trajectories? And can we intervene in high-risk cases to ensure that the language system give it the best chance to develop typically? For acquired disorders, we're asking questions like, in what ways can a language system break? Can the language system fix itself to some degree in cases when it starts malfunctioning? And what can we do externally to intervene on the brain to ensure that the language system recovers as much as possible? So we work on both developmental questions and questions of language loss in adulthood, and I'll give you a glimpse of the latter line of work. So this is a collaboration that we've had going for a few years now with Swati Kiran, who is at BU leading the Center for Brain Recovery, and the lead junior scientist is NBO, who is now a postdoc at Harvard. So understanding recovery from aphasia has been going on for many years. Centuries really now. So a lot of people have focused on trying to understand the trajectory, the timeline of development, and very consistently a lot of groups have shown that most recovery happens within the first few months, but in many cases recovery does continue even after the first year, although at a slower pace. Many other groups have studied questions like what makes some people recover better and worse. And one big finding is that the younger you are and the better your brain health is at the time the stroke happens, the better off you're going to be. But also it matters where the damage happens and how extensive it is. In our work, we've been trying to understand the recovery mechanisms that underlie recovery from aphasia. So to the extent that a patient recovers, what's happening in the brains of those patients? What are the changes that lead to the restoration of some of the linguistic function? And there have been two big ideas for how this might happen. One idea is what's referred to as homeostasis. So it's basically the idea is that you have some remaining parts of your language system, and they're trying to recover function by somehow reorganizing the function among the remaining bits. So you're trying to go back as much as possible to the way things were before the stroke event. A very different idea is reorganization. So this is a more drastic takeover of function by areas that weren't doing language before, but are now going to be doing language. And a good candidate for this kind of takeover is the system that many people call the multiple demand system or the executive function system. It's a very cool system you all have. It supports so-called executive abilities like working memory, attention, inhibitory control, and it's active across many, many different kinds of behaviors. One way to think of this system is it's a system that evolution gave us to be able to adapt, right? We have all sorts of specialized mechanisms like for hearing or for vision or for language, but we also have resources to be able to deal with change. And this system, the idea was that this system is flexible enough so that if some specialized mechanisms get damaged, the system can take over. Why does it matter? Well, if we know that it's a language system that helps you recover the remaining parts of the language system, or this very different system, we can intervene on these systems, right? We can either stimulate them or do behavioral therapy that targets this particular system. So it really matters for trying to boost the recovery mechanisms that are in place. So we try to evaluate these two ideas in a group of patients with chronic aphasia and a bunch of controls. So what we did was record their brain activity as they listen to language to understand which parts of their brain are processing language. And we also measured their linguistic ability in all sorts of ways using standard assessments. And so then we're basically asking, okay, to the extent that these patients vary in how good they are in language, what's different about how their brains process language so that some are doing better than others? And it turns out that it's really homeostasis that's doing this work. So it's the same system that was doing language in your brain before stroke whose activity is best predictive of how well you're doing after a stroke. So now, and individuals who show most typical like patterns in that system are the ones that do best. So what we're doing now is trying to evaluate a range of therapies, some based on behavior. So for a while, people got really excited about this idea of takeover of function, and a lot of therapies got shifted to trying to increase general ability, executive function ability, right? Let's make people better at working memory or at keeping attention in the hope that it helps restore these lost functions. Turns out that no, you need to really focus on language and keep training the language system up. We're also doing work with electrical stimulation of the language system, remaining parts, to see if it boosts recovery. We're also doing this very hard thing of longitudinal scanning, the very little is understood actually about the acute stage of what happens in the first few months, because most people don't want to do research when they just suffered a stroke. But it's really, really important to understand how things change during those first few months after injury, so we're trying that. And much like what Josh was talking about, we're also building computational models based on language models like ChatGPT, including creating kind of aphasic variants of these models to get a more mechanistic level insight into how things can break and hopefully how we can fix them. So thank you very much. Thank you.
Big chair.
Yeah. It is a big chair.
So we have time for some questions. Are there any that get us started off? Tony, use a mic back there.
In animal studies, does language have to exist for thinking to exist? This is something you presented. And I've read some studies that say that animals actually think even if they cannot speak. Is there a biological reality to that?
I mean, animals certainly do all sorts of complex cognition. Whether their thinking mechanisms are exactly the same as humans, well, probably not. But whether they're just whether humans have simply bigger, better versions of those mechanisms is potentially likely, and a lot of people are working on this. But yeah, you can definitely think without language, including in animals. Yeah.
Any other questions?
You mentioned something about language development with the very young, and you said around 3 years of age, things change. But previously I've heard a lot about much later than that, like learning a foreign language up to age 10, 11, somewhere like that. How do these two relate?
Sorry, I didn't quite hear everything you said. So how we learn later in life?
You mentioned in the talk that around, I believe it was the age of 3 years, that the way the language acquisition learning was taking place dropped off at that point.
No. I was just saying that the system I was talking about is there by age 3. Of course, learning can happen much later than 3. It's not,
Okay.
yeah, it still works.
Because I'd heard like 10, 11 years old.
Into the teenage years,
Yeah,
you can learn language in a native-like way, yeah.
I have a question. In terms of the scope of your research, do you also look into cognitive, I guess, impact caused by other diseases, such as cancer entering the brain, for example?
I guess some of the tools that we develop, we do work with brain cancer neurosurgeons. So people, this is not about cognition, but our microscopy methods, if you can find brain tumor cells more accurately. We had a paper earlier this year where we showed that you could get much more accurate counts of aggressive brain tumor cells with our new imaging methods than with older ones. So not exactly cognition, but the tools can be definitely applied.
Yeah, I was more referring to metastases of other cancer into the brain.
Yeah, I don't think any of us work on that per se. I'm very generally interested in what happens as people age. We do a lot of work on the consequences of hearing loss, and that is also accompanied with other kinds of cognitive changes. So that's sort of one thing that's a little bit related to what you said. And of course, there's things like Alzheimer's and Parkinson's that play into that as well.
So Mark, going back to your discussion about amblyopia, what's the best you can do right now for someone who has amblyopia?
Well, as I mentioned, patch therapy is the standard of care. The severity of amblyopia depends on the cause. So there are more benign causes, less severe causes that can be treated as late as age 7 or 8. So again, it's patch therapy. But in recent years, there's been introduction of what's called dichoptic therapy, which is using binocular visual stimulation, and they're getting very good results. But again, the effects so far are restricted to this early developmental period. So.
What causes a loss of language ability with age, hunting for words and so on? And are there ways to recover from that?
That's a great question. So I'm also very interested in aging, like Josh is. And we've just finished a study looking at how the language system in the brain changes with aging. And we can't find any ways in which it changes. So Neuralate looks just as good as a system in young brains. It's as strongly active, as strongly lateralized to the left hemisphere, responds similarly selective to language. And so there's no obvious changes there. So I suspect a lot of the word finding difficulties that people report with aging have to do with more general memory systems. And of course, those are very susceptible to disorders like Alzheimer's and in general decay with healthy aging. And those changes you can see very well even with methods like non-invasive imaging. I don't know if there's more to add.
If you don't mind, I have two questions. One is with the cochlear implants, do you find that there's more neuroplasticity in people who lost their hearing at a later age as opposed to people who never were able to hear? Do they adapt to a cochlear implant better?
Yeah, it's a little bit complicated. So, in general, the best outcomes happen with people who are implanted at a very early age, right? And one plausible explanation for that is that, in general, the brain tends to be more plastic at younger ages, right? But there is also evidence that there are benefits to having some hearing experience, and so that probably factors in as well. Yeah, that's a short answer.
My second question is about language. You talked about these centers of the brain are for all multi-language, bilingual, multilingual. When people lose language and become aphasic, do they always lose all of their languages, or can they lose one of their languages if they're multilingual? And how does that play into your studies?
That's a great question. So in most cases, yes, you lose multiple languages, but there are interesting effects where sometimes people report that the language that's not a native language you tend to preserve a little bit better. And we think we just recently got a glimpse of a possible mechanism for that. I always found this really confusing, not making sense, but one thing that we see is that native languages tend to be processed a little more focally. So they don't always engage this whole system. There's a core part in the posterior, in the left temporal cortex, that seems to really, really be important. But for all of your later languages, so for me, English is a non-native language, and English engages this whole network very consistently. And so if you have focal damage in the left temporal cortex, it is more likely to wipe out your native language, and the remaining parts may work better in the later learned languages because it's just more consistently recruited in this way.
Oh, fantastic.
Hi, thanks for sharing your research. They are fascinating. I'd like to know if there's any studies showing the impacts of the early and frequent use of technology, social media, and generative AI by children and teenagers in core cognitive functions, because like attention, learning, memory, because we've been hearing a lot about the impacts of social media and screens in the mental health, like increased levels of anxiety and eating disorders in girls. But what about the other functions, like using technology can harm in the long term our attention, language acquisition, memory learning? Do we have any robust evidence on that? Thank you.
Yeah, to my knowledge, there isn't actually robust evidence one way or the other. I mean, there is a literature that, I mean, Ev may remember this better than I do, but there were a lot of studies for a while showing that you could see some advantages from people that played a lot of video games, like benefits on visual attention, for instance. And I'm not up to date on what the current status of that is. Did that stuff hold up? I can't remember.
Yeah, and that research program on video games started because people thought that video games are destroying our children's brains, and they started looking at all sorts of functions like attention, memory, and it turns out everything they looked at is actually a little bit better. So that's where that went. And yeah, I have not heard of any research other than the mental health effects of exposure to social media, that it affects attention or memory in any negative way, or language.
I still keep my kids off it.
Hi, thank you so much. The brain is so amazing because there's still so much we don't know. I've heard about how people are trying to, I mean, I guess all of you guys in your own way, but are trying to map like each circuit. And to me, it sounds like the kind of impossible dream that we once thought mapping the whole genome would be. So do you think that's something that we'll see in any of our lifetimes? I mean, just understanding how all the different pieces fit together, or is that like way out there? Thank you.
I think everybody should take a shot at that one.
Go ahead. Me? Yeah, well, I think one of the great things about being here at MIT is the technology collaborations, right? So great engineering school, great science school, great crosstalk, and the influx of everything from nanotechnology, from neural interfacing to new non-invasive strategies for delivering energy and recording information from the brain. I think that's presenting a huge amount of almost engineered serendipity between different fields that is opening up a lot of a bit of a Moore's law in terms of improving our ability to look at and control brain circuits. So being an engineer, maybe it's how I think about things, but I think being at MIT, we're surrounded by people who are always asking for what the next big problem is that they could work on. And they may have revolutionized one field, and they're looking for the next big challenge. And for many people, that's the brain.
Mark, any thoughts?
No thoughts.
Okay.
No. I mean, I think there's value in simplification. I'm a lumper, not a splitter, and looking for general principles seems to me a lost art that needs to be reinvigorated, and I hope to see that continue.
Yes, I mean, one thing I would say is, I don't know how realistic it is to think that we will actually understand the human brain fully at the circuit level. And I'm not sure that we will necessarily need to for a lot of the things that we want to do. So for instance, like if we end up really understanding learning, right? And if learning is the key to a lot of the things that we want to do, then that may be sufficient. And having really detailed explanations of what happens at the circuit level, may or may not be critical for what we actually want to do.
Oh,
So I think there will be examples where we have really circuit level explanations for things, but I'm not sure that will generally be the case for the human brain, just because it's so big and so complicated.
Hello. I'm from bioengineering department, and we do run the clinical trial with the long COVID participants and chronic Lyme participants. And I've been always very fascinated about the patients complain about their brain fog symptoms as #1 major symptom complaints. And that's why I'm here. And I got very fascinated with the language aphasia presentation. Thank you so much. And I would love to, because I don't have much basic knowledge in the brain science, but I want to ask this question for my patients actually here, that this is all new symptoms in terms of being lost in the middle of the speech when they're trying to engage interaction with someone in conversation. And the way they describe brain fog and cognitive deficit is oftentimes related to their language difficulties. And I would love to know your insight in terms of the brain function associated with the infection triggers or source. Not so much a stroke issue, but perhaps what are the insights with the infection triggers, and can this kind of a brain fog complaint description of language difficulty be something to look at in the Wernicke's or Broca's area, or what are the insights that we can look further to expand our research? I would love to hear your feedback. Thank you.
Yeah, I don't know if I have too much to say about this. I mean, from my understanding, the brain fog-like symptoms in long COVID patients are not quite specific to language. Language may be affected, but it seems like it's a pretty widespread systemic effects on multiple systems. So it's possible that insights from specific disorders like language or audition-related things may be helpful, but it seems like there's some more global things to understand there. And maybe the deficits are actually in this very vulnerable system to aging, which we mentioned, the system that affects memory and is very affected in Alzheimer's, so the system that has big hippocampal components. But yeah, sorry, I don't think I have anything more specific than that.
No, thank you so much.
So a question for you before you leave. So what do you measure to sort of document the patient's brain fog? What kind of tests do you have them do?
So currently in our study, we do 3 different measures that we add. So typically a day before a person comes to MIT visit, for study visit, we assign the cognitive test called brain check. It's an equivalent version of a battery that can be done remotely. So it takes strobe and trail making and memory function, immediate recognition type of testing. And once they come to MIT for study visit, we do 2 different neurocognitive testing. One is called right eye, which is we add a visual sensitivity testing. So the person will we're gonna look at the eye coordination and pursuit and saccades and the brain reaction time and physical reaction time by using different images and how quickly they react. And the other one, we also use equivalent version of the EEG called WAVI. It's actually a commercial testing, but we look at the brain activity, look at the alpha peak wave during the P300 testing. We also do the flanker testing as well, just to see the difference in terms of reaction time.
Great, thanks. We can learn from the audience as well.
So, hi, I'm Shupeng, and I'm a first-year master's student at Harvard. So my question may be a little bit naive. So in my undergrad study, I did projects related to, understand the molecular mechanism of autism. And I also have some experience dealing with the neural image. So my question would be, well, it is possible to have a model, like machine learning model, to actually predict both the genomics and the outcome of diseases, or the hearing aid devices for both genomics level and also the circuit level. So if that is possible, so what is the challenge that you may see in this,
So we have less than 2 minutes.
You want to try that?
Josh, that's you.
So, we're definitely not there, right? I think the means by which genes lead to circuits, that's a pretty big and hard and complicated problem. I don't know. I mean, machine learning could certainly be useful probably in understanding that if you had enough data. And my guess is there probably will be lots of headway on that. I mean, there's definitely lots of people who are very excited about building models of like the entire brain sort of at the circuit level. I mean, this is sort of, I think, what Ed maybe aspires to. And you know, where that's going to lead, I think we still don't know. But it's a really exciting direction, and you know, it could become within reach.
Okay, thank you.
So one last question.
Thanks for your talk. I'm curious, I have actually two questions, if I might. With regard to language, are the same centers for language processing identical regardless of the input via hearing, sight, signing, etc.?
Yes, yes, sorry, I didn't explicitly say this. They're amodal, so it doesn't matter if you're reading or listening or processing sign language if you're sight.
Okay, my second question is, how broad is the definition of language? Does it encompass situational awareness when you're in traffic lights, where cars are, danger on a street? Is that part of the language function?
Well, so I certainly wouldn't put, I mean, language is a very difficult construct in the sense that it interacts with all sorts of other systems. But it so happens that we have a set of these areas that are quite specialized for language. And then a lot of ongoing work is trying to understand how these language circuits work with low-level audition circuits, how they work with memory circuits, how they work with attention circuits. Because of course we use language in its real-life complexity where there's a lot of systems that come together and have to talk to each other. But it does seem that there's some sense in which there's a system that's specialized for just this encoding, decoding aspect between thoughts and word sequences.
I guess the follow-on to that is if I lost my language centers, would I lose my ability to understand situational awareness?
No, you would not. In fact, there is a large body of work with severe aphasics, and their cognition, so it relates to the first question as well, their cognition is totally fine. They can navigate the world, they can socially reason, they can keep their attention, they can play chess. They just can't express their thoughts in language or understand others.
Thank you.
So to be in sync with the remaining part of the program, let's thank our speakers. And I guess they're going to reconvene across the way there very shortly.